So in this video, I'd like to talk about DNA sequencing and some of its applications. So the first thing when you learn about DNA sequencing, the first thing to know is that we really live in an era of remarkable growth in DNA sequencing technologies and availabilities of DNA sequencing. So what we're looking at here is, so this is a graph of the number of kilobases, that's thousands of bases, that a sequencing machine could read per day. So down here, with what was called a manual slab gel machine in 1980, you could get about 10 kilobases, about 100,000 nucleotides per day. Whereas now, we are sort of out here, we're in the future. What you can see is a single machine now. This says a billion nucleotides, but it's closer to a trillion nucleotides per day per machine. Another way of looking at this is the cost per genome. So this would be the cost per human genome. So here we have the publication of the first human genome. It was about $100 million to make the first human genome, and was published in 2001. And then what we can see is over time this declines. And notice that, again, this is on a log scale, so that this isn't just going down slightly. This means that within about 7 years, the cost of sequencing had gone down tenfold. And then what you notice is that it was going down fairly quickly for a while, and then it started just going even faster. So that now out here, we are rapidly approaching when we could sequence a whole human genome for 1,000 nucleotides. And we can actually compare that to Moore's law. So Moore's law is an observation about computing power that said basically that the density of transistors, and therefore the speed of computing, goes down or doubles every 2 years. And so what we see is that sequencing was basically following Moore's law, that going down by a factor of 2 every 2 years, and then it started going much faster. And here are a couple other graphs that I'll put in the notes for you all to look at. Here in particular, so this is the number of named sequences, so sequences where we have some idea what they are. So you can see back in 1985, there were about 1,000 available in public databases, and now we're well over 1 billion of these. So there are two major forms of sequencing that we're going to talk about. The first was Sanger sequencing, and so this uses an approach called chain termination, and on the next slide, there's a link to a video if you want to learn some more about that. The way this works, so this is targeted sequencing. So what we're going to use is PCR, which we'll learn about in another video, to amplify a single product, a single gene that we want to sequence, and make many copies of it. And then we can sequence this product. So this kind of sequencing is high quality, has very few errors, but it's quite expensive. So this won't sound expensive, but $1 per 1,000 nucleotides compared to next generation sequencing is quite expensive. And so here's the link if you want to check this out. But what we're going to focus on today is massively parallel next-gen sequencing, which are the modern technologies that are used now widely to study everything from ecology to human disease. And so here's a link to the video. I'm just going to play this video, and we will and I will guide you through it. Okay, so we start out with really long, big pieces of DNA. So it turns out, as we'll learn, that we need DNA to be the DNA that we're going to sequence to be particular lengths. And so what we're going to do is we're just going to randomly fragment that DNA. So here they're doing it with sound. There are multiple ways to do it, but we're basically going to randomly fragment our DNA. So instead of having huge, long chromosomes, we just have little chunks. So the next thing that we're going to do is we're going to add these adapters. And these are like grabbers, like on the end of corn in the cob when you stick it into the ends of your corn. So the point here is that these are going to allow our machine to actually physically grab a hold of these sequences. So we're going to ligate these adapters onto the end. So the next thing that we're going to do is we're going to run this out on a gel. This gel separates things by size because we want all DNA fragments that are of the same size. Okay, so where we're at right now is that we have fragmented our chromosomes into small pieces and then added these grabbers to the end and then selected pieces that are all the same length. Okay, so now where we take these DNAs and we turn them into single-stranded DNAs. Now all this stuff you're going to be seeing here, these are basically tricks to add additional grabbers. So we've got a grabber on the end that allows this, basically this is a single-stranded piece of DNA that is complementary to our grabber. This is why we needed a grabber. And then this is the DNA polymerase, which is going to take this and extend this strand so that we'll have a double-stranded DNA, but we've added this little chunk of DNA which has a particular sequence, and we're calling it P7. Then we do it on the other end with something we call P5. And so now we'll just turn that into a double-stranded DNA. So, okay, so all we've got here is, so we have our sequence of interest. This is the actual biological sequence that we're adding, that we're sequencing, and then we've added these grabbers on either side, and then we've added these other bits. And these are sort of a second round of grabber. We'll see why these are important. Okay, so now here are our double-stranded DNAs with our extra grabbers on the end. And as these fly, so what we're looking at here, this is a glass slide. It's called a flow cell. So it's a glass slide that has all these adapters on it, and these are things that are able to bind to the particular grabbers on one end of our DNA, the P7 or the P5. And so we basically just at some density we let these DNAs flow across the cell, and some of them will stick, right? So we don't want every single one of these things to stick, but every once in a while we want one of them to stick. So once it sticks, so that now we've got different DNAs. These are independent DNAs from our sample that are here and there on the slide. Now we're going to add the polymerase to make them double-stranded. Okay, so now what we have is one DNA, and we're going to, as we'll learn soon, we're going to sequence this DNA by adding different nucleotides. And every time we add a different nucleotide, it's going to fluoresce. So basically the strategy is going to be we add a nucleotide and it's an A, and so it's going to fluoresce green because let's say A is green. And then we'll have a camera that'll say, oh, green, there's an A in this nucleotide. And then it'll be G, and be like, oh, okay, this is G. All right, so that's the way this is going to work. The problem then is that just a single nucleotide being added to a single strand of DNA does not make enough fluorescence, does not cause enough light to be detected by our camera. So in this next step, what we're going to do is we're going to amplify our one strand of DNA so that we make, or sorry, our one piece of DNA so that we make many, many pieces of DNA that are the same. And then, so what these adapters on the slide are doing is just keeping everything local so that we build up a team here of a bunch of different DNAs that are all identical to each other. So now, when an A gets added to every single strand, every single one of them will be emitting a little bit of green light. So it's kind of like we're building up our local cheerleading squad so that they can all scream A together and then C. So all we're doing now is we're basically, so this one strand of DNA got turned into two strands by the DNA replication machinery, and then the other end got attached. And then these two strands were denatured from each other, so now this is one strand and this is the complementary strand. And now we're going to, so this is a bunch of rounds by which we basically take one strand of DNA, turn it into two, then curl it over and attach it, and then denature it. So we have a single strand, we turn it into two. So let's do that again. All right, so we've got one strand. Oh, let me go back one bit more. So we've got a single strand. Use a polymerase to make two strands. These two strands get bent over, so different, and now they separate. and different of our two strands are attached to our adapters. Okay, and so now they get replicated, and then they bend over again and separated. Replicated, bend over again, and separated. So now what we have is all these DNAs are here we have many copies of the same DNA. Here we have many copies of a different DNA. Here we have many copies of another DNA. And so we add these nucleotides now. These are modified nucleotides, just a chemical trick where we have an A nucleotide that when it's added, it fluoresces a little bit. And we've got a C that when it's added, it fluoresces a different color. And G that fluoresces a different color. And so now we can just use a camera to capture what's going on. Right, so here is showing how one base gets added at a time for each one of these nucleotides. So basically, we've got all these different DNAs being created, each one fluorescing a tiny little bit. And so you get a pattern like this with different spots giving you different sequences. Okay, so that's just a little bit about this technology. So now that was to give you a sense of how the actual chemistry works. And so now let's actually talk about what this looks like if we're trying to sequence a whole cell, or all the DNA. I shouldn't say a whole cell, but all the DNA in a sample. Okay, so here let's imagine we have 3 chromosomes, chromosome 1, chromosome 2, and chromosome 3. So we said we're going to fragment them, and then we're going to sequence each fragment. So if we do just a tiny little chunk to give a sense of what this would look like, so that would mean that we fragment the DNA, and then we're going to get 3 different, we call them reads, short sequences. So one would be TTAC, ACT, and then the other one would be AGAC, and so forth. So if we color these 3 different sequences, we can recognize that by just having these 3 sequences, yeah, well, we know these 3 short sequences, but we don't know what order they go in. So it could be the red one, and then the purple and the blue, or it could be the purple and then the red and the blue, or whatever. So we need to have some way of ordering these sequences. And it turns out that if we sequence if we sequence this chromosome again and again, well, it's going to get fragmented in different places. Right? So this is identical chromosomes that we fragmented independently, so we get fragmenting in different places. And so if we just, for instance, look at 2 of these, what we'll see is that we get these overlapping chunks, right? So this CAAC TGAG is this CAAC TGAG. Okay, so we fragmented it in different ways, so what we end up with is just a pile of reads from these 2 copies of the same chromosome. And we don't know their order, but we can actually use their overlaps to stitch things together, right? So we can recognize that this CAAC TGAG is found in this read and also found in this read. So that means we can put them together. And so now we have a slightly longer sequence. And then we can recognize that this overlaps with that, and that this overlaps with that. And so we can use these overlaps to put our sequences in order. And so we can end up with the assembled sequence. We call this genome assembly, because we get this larger assembled sequence. Okay, so there are complications. So we can have real SNPs. That would be, I made a mistake here, but this should be the A and T. So that this would mean 2 copies of the same chromosome that are different because of differences in the sequence, sequence mutations between the 2 alleles. And you can also have errors in sequencing. So this is a little bit of a complication, but people have built complex programs to deal with these problems. So to give a sense of the scale, so here we're looking at genome size. So viruses are between a few thousand and almost a million nucleotides. Bacteria tend to be on the order of a few million. And then vertebrates are up here with a few billion nucleotides per genome. So this is the amount of DNA in a single cell. So Illumina sequencing, so Illumina is the technology that we saw in the video. So it currently produces reads that are on the order of 100 or 200 nucleotides. So that means then we have to have 20 million different reads just to cover the haploid human genome once. And to do sequence assembly, we want to do maybe about 30 times. So we did 2 when we showed the stitching together by overlaps. but you actually need more than that for various reasons. And so that means to sequence the human genome, we would need about 600 million Illumina reads. Fortunately, it's quite cheap. It's about $1,000, maybe even cheaper now, for about 50 million reads, which would cover the human genome twice. So that means that even with things, but you can see here, even at the price that things currently are, which is quite cheap, it would still cost several $1,000s to independently sequence a human genome. So fortunately, if you already have a genome, you can use the reference genome to assemble your reads. So we've talked a little bit about differences and similarities between us, but for the most part, different humans are about 99.9% identical. And so what that means is that if I have your genome, or if I know your genome sequence, then I almost 100% know my genome sequence, because 99.9% of it will be the same. Obviously, the differences are important, and that's what we want to find, but it means that we have a lot of information already. And so what we can do, we can take what's called a reference genome, which is it's not actually from any single human, it's kind of a combination. And so this is a genome here we have chromosome 3 that has been well assembled. We're very confident in the sequence. So then we have my genome, Scott's genome. It's been broken up into chunks, and we've sequenced individual reads. And so we don't know the location of these reads, but what we can do is take individual reads and map them to the genome. So we basically take this read, we take the sequence, and we scan all along chromosome 3 and try to find where the similar sequence or identical sequence is. And we find, let's say this one goes there, and we find this one goes there, and we find this one goes there. And so we can take all these reads, we can map them to the genome, and it just greatly speeds up assembling my own genome and makes it a lot cheaper. So this would be what this looks like at a sequence level. So here we have my reads and then the reference. So we can take this one and we realize, oh, it's almost identical there. This one's almost identical there, almost identical there, and so forth. So one by one we can map them. And then we can combine this into what my genome looks like. So that would mean that basically we find that my sequence goes like this. So sometimes we won't know because no reads mapped these two nucleotides, and no reads mapped here. But it can give us a lot of information about differences between me and the reference. And so I should point out, I've simplified this here. We would expect that maybe I would have a SNP myself, a difference myself, where I'd have two chromosomes, my two alleles. Maybe I'd have an A and a C here, and maybe I'd have a T and an A here. So I've simplified things, but this is the general strategy for identifying differences between me and the reference, or me and you. So another thing that we can do is we can use DNA sequencing, Illumina sequencing, to study RNA expression. So this is called RNA-seq. And so the way this is going to work is that instead of taking all the DNAs and grind them up, we're going to take the RNAs and then convert them into DNAs by a process called reverse transcription. And then we're going to map these to the gene sequences to determine the level of gene expression of different genes. Because as we've discussed, gene expression will be different in different cells, and it can be very high, it can be very low, it can be on, it can be off. So to study this, so we can take RNA from my tissue and then perform sequencing of it. So we get all these RNA-seq reads, and now each of these we can map to the genes in the genome. So here I'm just showing 4 genes, but if we did this, it would be to my 25,000 genes. And so what we see is we have some genes that have a lot of reads that map to them. So that would mean this gene is very highly expressed. We have some that are medium expressed, some that are low, and some that are not expressed. Right? The fact that that in our sample we have no reads that map to this gene suggests that this gene is not expressed, or only at a very low level. And so a major thing that we can do then is to understand the differences between tissues, or the differences between individuals, or the differences between people suffering from a disease or not, is we can compare between different samples. So here this would mean we take a brain sample and we do RNA-seq on that sample, and then we do the same on a liver sample. And then we have our 1, 2, 3, 4 genes, or 1, 2, 3, 4 genes. So this would be the same gene 1, gene 2, gene 3, gene 4. And then we can compare. And so we can identify genes that are probably involved in brain function because they're expressed in the brain and not in liver. So these are probably related to specific brain functions. Then we have genes that are only expressed in the liver and hardly at all expressed in the brain. So those would probably be related to liver functions. And then we have other genes that are expressed highly everywhere, other genes that are expressed lowly everywhere. So this is the way that we can start to understand how differentiation of gene expression is involved in differentiating tissue function. And so just to give an example of the kinds of questions we could ask here, so how many genes show clear expression differences between brain and liver? And so we can see that there are basically two, gene 1 and gene 4. So here's a question to think about. So using techniques that we've just discussed, how could we use genes that are regulated? How could we study genes which are regulated by a transcription factor? And so you can think about this for a moment if you want to pause the video. So one way we could do this, we could compare samples. One where we have a wild type that say has some transcription factor, has some protein that binds to the gene and binds genes and influences their regulation, and then a sample that does not have this. So we haven't actually talked all the way out, but there are ways that we can knock down genes. So we could have two samples, one which contains the gene that makes this transcription factor 1, and one of which doesn't. Okay, so main difference here is that the wild type will have this transcription factor and the knockout will not. And so so if we do that, we can do the RNA sequencing and we can start to identify some differences. So for instance, here gene 1 is expressed in the wild type, but then it's not expressed in the transcription factor knockout. This one is the other way around. It's expressed in the wild type and not expressed in the transcription factor knockout. So let's focus on these two genes. So we can ask, the data suggests which gene is positively regulated by transcription factor 1. Okay, I'll give you a second to think about that. Go ahead and pause if you like. Okay, well this one, so remember here we have transcription factor 1, and here we do not have transcription factor 1. So this gene is expressed when we have transcription factor 1, but not expressed when we don't. So that would suggest that transcription factor 1 may be positively regulating this gene, and may, so that the gene is only expressed when it's present. By contrast, over here, when we subtract TF1, we find that the gene is more expressed, or another way to put it would be when we go from the knockout to adding in transcription factor 1, that the gene goes down. So that might suggest that transcription factor 1 is negatively regulating this gene. So another way that we could do this, that we could study where transcription factor 1 are binding and influencing gene regulation, would be to use CHIP, which is chromatin immunoprecipitation. So this is basically, it's similar to this is similar to the antibody methods like Western blot that we discussed, but it's a little bit different. So the way that this is going to work is as follows. Okay, so we've got our transcription factor. We're going to raise an antibody to it. So now we've got this grabber. So now when we add this antibody to our cell, it's going to go and bind the transcription factor wherever that is binding to the DNA. So now we can cross-link the DNA and the protein. That just means cause covalent bonds between the DNA and the protein so that it'll stay together despite the fact that we're going to disrupt the solution in various ways. And so then if we fragment, so now what we can do is we can fragment the DNA. So it gets all into chunks, and we're going to degrade anything that's not linked. And so that means all the stuff in the middle that is not linked to our transcription factor we're going to get rid of. And now we're left with our transcription factor binding to a specific chunk of DNA. And so remember, this isn't just we're representing this as a line, but this sequence is different from this sequence, right? This is the coding sequence for the gene, and this is the sequence right next to it, and this is some other sequence. But if we looked in, this would be ACGT, or whatever, and this would be a totally different sequence, ATG, TTTTTT, AAAA. So these different pieces of DNA are identifiable as specific chunks of our genome. And so now we can collect these chunks and sequence them and then map them back to the genome. And so then what we get is we get these reads that show where transcription factor 1 is binding. Right? So the fact that we get these reads when we do this process tells us that transcription factor 1 is binding here, but that it's not binding here. So we can also do a similar thing. So this is called Clip-seq, and it's a very broadly used method for understanding where transcription factors bind. So we can do a similar thing with RNA binding proteins, like, say, proteins that are involved in splicing, and that's called Clip-seq. So that's all I'd like you to know. So we can also do it on, if we want to figure out where the ribosomes are binding, so to look at rates at which different mRNAs are being translated, we can do something called Ribo-seq, which is similar to Clip-seq. So this tells you where the ribosomes are binding. This allows you to find RNAs that are more efficiently and less efficiently translated. So then we can look at differences in rate of translation, regulation of DNA translation, and how knockdowns of different RNA binding proteins affect RNA translation. Okay, so the kinds of questions I'd like you all to be able to answer are like this. How do we find SNPs? So we said that we do that by doing DNA sequencing. We sequence the DNA and figure out where there are differences, polymorphisms. How do we study gene expression level? We said we'd use RNA-seq. What's the most direct way to study which genes are regulated by transcription factors? So there we're looking for the answer Clip-seq, because we did say we could do it with RNA-seq and knockdowns, but Clip-seq we're really directly looking at where the transcription factors are binding. Similar thing for RNA binding proteins. We said the answer there is Clip-seq. And what's the most direct way to study rates at which mRNAs are translated? And so that's Ribo-seq. Okay, so these are basically the 5 things, kinds of questions we want to know. We use DNA sequencing to study differences at the DNA level, RNA-seq to study gene expression, Clip-seq and Clip-seq to study the binding of DNA binding proteins and RNA binding proteins, and Ribo-seq to study translation.